Okay, in chapter 5, we're going to continue with kind of where we left off in chapter 4. In chapter 4, we were looking at parts of cells and what those parts kind of do in general, and we're going to kind of continue in that vein, but we're going to spend quite a bit of time talking about membranes of cells and what they can do, how they help keep the cell interior different from the cell exterior, and really, once again, we're going to see that these membranes, their structure, is so critical to their function. We're also going to move on into how energy is used in cells, transformed within cells, and how that energy really gets work done. And we're going to see that important players in getting work done within cells will certainly be enzymes. So that's where we're headed. Now, in this diagram, what we have is we have an example of right here. We have, this is one plasma membrane. This would be the interior of that cell. Here is the extracellular matrix out here. Here are those collagen fibers we talked about in the previous chapter. Here's a fibronectin that's connecting a collagen fiber to an integrin. And we also have some of these carbohydrate and protein fibers out here in the extracellular matrix as well. Now also notice that we have here some integrins. We have a peripheral protein. We go over here, and here is one of the integral proteins that is capable of binding a signal molecule, and then that way that would lead to really the cell being notified of a way in which it should change its behavior in a process called signal transduction. So here we see different integrins doing different things. Here's another integral protein. It's a channel protein, and you can see it's allowing some things to, and actually it looks like an aquaporin, which is allowing water to come into the cell. Remember, water's polar. Water has a hard time getting past the hydrophobic zone, and so the aquaporin, the channel protein, helps water get across, shielding it from the hydrophobic zone of the bilayer of hydrophobic tails. We also here have a carrier protein, another integral protein, transmembrane protein, but in this case we see that there's this molecule or ion or whatever it is binding to the binding site here of the carrier protein, and then we have to add ATP to that protein to get it to change its shape so that that molecule or ion is moved outside the cell. Here we have another integral protein that is binding to this integral protein of a second cell. This is a second cell. It's membrane. This would be the cytoplasm of that second cell. And here we have membrane proteins helping tie together adjacent or neighboring cells. Now down here we actually have a glycoprotein. Here's the protein part. Here's the sugar part coming off that protein. And this neighboring cell has a membrane protein that is recognizing, checking out the shape of that glycoprotein. So if we go up to this other cell, we see it has some different integral proteins. It has a couple of adjacent integral proteins that are carrying out a reaction. They're enzymatic in their nature. They're the first member of the pair takes on a substrate, converts it into an intermediate, which is acted upon by the second enzyme, which gets the final product. Now let's see. So anyway, we're going to be looking at cellular membranes and how some of the jobs that they do in terms of allowing certain things in and out of cells. Now, one of the things we mentioned here in the headline is that membranes are fluid mosaics of lipids and proteins with many functions. So we've seen there are many functions to the proteins that are embedded within plasma membranes. But also membranes are fluid mosaics. Now, a mosaic is a larger picture made up of tinier pieces, smaller pieces. And that's what we have here. We have different kinds of integral proteins. We have the phospholipids that are helping make up the membrane. And also right here and right here we have cholesterol, which is also a part of making up membranes as well. Now, when we say that they're fluid mosaics, what that references is that these phospholipids can actually slide laterally past one another. And actually, some of these proteins can actually move through the membrane as well. So fluid mosaic, stuff can shift around a bit, and that involves lipids, and it also involves proteins, and once again, many different proteins carrying out many different functions. One of the things we have got to keep in mind is that these selectively permeable membranes are monitoring what moves in and out of cells. If something has a charge about it, if it's polar, if it's ionic, they're going to need help getting across. That's what we're seeing here with the aquaporin in water, is that water needs some help getting across, and so there is a protein called an aquaporin that can help water get across. Here we have probably an ion that needs help getting across, and so once again, proteins oftentimes are involved in this. We've already noted that phospholipids can slide past one another in a lateral fashion. That contributes to this idea of membranes being fluid. Also, something else that's kind of interesting is that as seasons change, as environments change around us, sometimes we're going to be changing out our phospholipid tails. Sometimes we'll replace them with some, as temperatures get cooler anywhere, environments are colder, oftentimes we'll change out some of them to where they have a kink. There's a kink in those phospholipid tails, and that kind of helps offset, loosen up the membrane because the membrane's experiencing colder temperatures. Cholesterol is also critical to maintaining appropriate fluidity in membranes as well. So a lot of times we think of cholesterol as just being a bad thing, but it's not all bad. It's just too much of it can be a risk factor. Well, we've already noted there was quite a bit of protein diversity in those two cell membranes that we looked at on the previous slide, and we see that same picture here on this slide. We did point out the enzymatic proteins. Oftentimes they are placed in close proximity to one another, like we see here, so they can carry out sequential reactions. We've also pointed out that some membranes will actually form these junctions, these intercellular junctions that help hold neighboring cells together. We've also noted that sometimes, like glycoproteins, are actually kind of self-identifying. They're like an ID for a cell, saying this is a self cell, not a foreign cell. So oftentimes glycoproteins kind of serve as identification tags. Transport proteins we've already talked about. They help materials move across the membrane because the material might be too large or might have charge or something like that. And some of these transport proteins are going to be channel proteins, and we see that with the aquaporin here, but then some will be carrier proteins. Now, in the case of a carrier protein, keep in mind, the ion or whatever it is actually has like a binding site for it to bind to, and then in the case of active transport, that refers to the addition of energy to get the protein to change its shape so we get the material moved across the membrane. Receptor proteins here is what we're looking at in this area of the membrane, can receive and respond to signal molecules, and that can lead to something called signal transduction, where the signal ultimately can lead to maybe activation of an enzyme or the turning on of a gene or turning off of a gene, something like that. Now, we've already also looked at in the previous chapter, and here we've mentioned it again, that some of these integrins are actually tied to, like your fibronectins, which are tied to the extracellular matrix. And so there's really this connectedness between the extracellular matrix and actually the internal components of the cell. Now, when material is being moved across a membrane and no energy is being invested, the term we apply to that is passive transport. Now, one of the things we have to keep in mind is that molecules are in constant motion because they do have thermal energy. The only time a molecule is not going to have thermal energy is at absolute zero, which never occurs in nature. So molecules are in constant motion because of the thermal energy. So down here, what we look at is we see these molecules of dye. They're constantly vibrating, bumping into one another, ricocheting off, and by chance, some of them ricochet off at an angle that enables them to go across this selectively permeable membrane. So here's a molecule, it ricochets off of another one, it's going through the pore to the other side. So what we have is we have thermal energy, sometimes we call it intrinsic kinetic energy of molecules. They're causing them to bump into one another, they ricochet off, and some of them make their way from one side where it's crowded to the other side where it's less crowded. Now once again, this is an example really of passive transport or diffusion. Because there is no energy being invested by the system itself. It's just the thermal energy of the molecules that enables them to move from where they're crowded on the left side to where it's less crowded on the right side. So what we have then is we have molecules moving from the left to the right. Now, as some molecules get over here on the right side, it doesn't mean they're going to stay there necessarily. They too will bump into one another as conditions become more crowded on this side. And by chance, some of them will ricochet off and actually go back to the left. But because there are fewer of them, there will be fewer going from right to left or from the right side to the left side. Because it's more crowded on this side, you're going to have more going from left to right. So the net diffusion, the net direction is left to right. In time, there will be pretty much an equal amount of molecules of dye on the two sides of the selectively permeable membrane. Now, this is called dynamic equilibrium because the molecules will still be bumping into one another because of their intrinsic kinetic energy, their thermal energy. They'll ricochet off, but because they're pretty much in equal numbers on both sides, you're going to have as many going right to left as you have going left to right. So, once again, because of this intrinsic kinetic energy, this can lead to diffusion, whereby molecules are moving down their concentration gradient, which means, like we started off over here in this first part of the diagram, the molecules will be spreading out from where they're crowded to where it's less crowded, from an area of high concentration to an area of low concentration. And this will continue until dynamic equilibrium is reached. Now, this is really how oxygen moves from our blood into our cells. We have oxygen is concentrated in the blood, and then oxygen has an opportunity to move from the blood where the oxygen's at high concentration levels into our cells, which are experiencing low oxygen concentration. And it's the same for carbon dioxide. By way of cellular respiration, we're going to be producing carbon dioxide in our cells. And the carbon dioxide diffuses out of the cell, ultimately into the blood, down the concentration gradient. So we are relying upon diffusion, passive transport, to get oxygen into our cells and to get carbon dioxide out of our cells. Now, oxygen and carbon dioxide, if you remember what we said in the previous chapter, they are nonpolar. So they're not going to have any real trouble moving through the hydrophobic zone of the plasma membrane. Now, if something is ionic, like we said in the last chapter, or polar, then they're going to need some help getting across the bilayer. Now, whenever something like oxygen and carbon dioxide can move through the bilayer without any real trouble, we call that simple diffusion. They don't need any helpers. They don't need any proteins helping. But when you have something that's ionic or polar, they're going to need some help. And so, even though they're going to move down their concentration gradient without an input of energy across a membrane, that's diffusion, the helpers are going to be proteins. And so we call their movement facilitated diffusion because they have helpers. They have proteins helping them get across. These proteins will be the transport proteins like the channel proteins and the carrier proteins. Now, we can also talk about the diffusion of water across a membrane, and that's what osmosis is. What we have over here, we have this leftmost U-shaped tube, and then we have a right U-shaped tube. Now, we have a left side to this U-shaped tube, we have a right side. And when we look on the left side, what we see, another thing I should mention, I'm saying there's a left side and a right side, which are divided by this selectively permeable membrane. So water can move through the membrane, but these solute molecules, they're too big, they can't get across. Okay, now it's really obvious that, we have this U-shaped tube divided in half by a selectively permeable membrane. And what we have on the left half, if you look over here on the left half you see there are way fewer solutes as compared to the right half. Well, since the left half has a lower concentration of solutes as compared to the right half, then what we're going to say is that the left half of this U-shaped tube is hypotonic relative to the right half. Okay, so whenever there's a lower concentration of solute, the term for that is hypotonic. Now, this right half has a higher concentration of solute, so we're going to use the term hypertonic. So whenever we use these terms, hypotonic and hypertonic, these are relative terms. These are just comparison terms. So the left side is hypotonic, the right side is hypertonic. Now, if you notice though, the two halves have equal volumes. But in the left half, the volume is consumed by by more water molecules and fewer solute molecules. The right half's volume is consumed by more solutes and fewer water molecules. Okay, so when we use the term hypotonic, it refers directly to there being fewer solutes, but indirectly to there being more water molecules. Hypertonic refers directly to there being more solutes, but indirectly refers to there being a lower concentration of water. So, hypotonic, lower concentration of solutes, which also implies, of course, a higher concentration of water like we see in this drawing. So the right half, as we said before, is hypertonic relative to the left half. Now, what we have here is this line box, and it gives us some more detail. Here we have water molecules. The red part of the drawing represents oxygen. The two little kind of white gray are hydrogen, so H2O. And you notice there are fewer solutes that the water's attracted to. Let's say this is a sugar, and the water molecules are attracted to the OH groups on the sugar. Well, there are a few sugars on the left side, like we've already pointed out. So that means there are fewer water molecules being tied up by sugar. That means there are more free water molecules. This is a free water molecule, this is a free, this is a free, this is a free. All of these are not free water molecules. So there are more free water molecules. They have intrinsic kinetic energy because of their thermal energy. They bump into one another, they ricochet off, and by chance they can get through the pore of the selectively permeable membrane. Now over here on the right hand side of the U-shaped tube, this first U-shaped tube, we see there are more solutes tying up more water molecules, so therefore there are fewer free water molecules as compared to the left. Now these few free water molecules, they will bump into one another. By chance, there will be some moving back across the membrane to the left from right to left, but it's not going to be near what we're getting moving left to right, because on the left side there are more free water molecules bumping into one another as compared to the right side. So more free water molecules on the left side versus the right half. Now, we've already mentioned that in time what's going to happen is that the water molecules will continue moving from left to right. There'll be some coming right to left, but the net movement will be left to right until dynamic equilibrium is established, and that's what's being shown in this right U-shaped tube. The two halves of the U-shaped tube are unequal in volume, certainly, but they're equal in tonicity. They're equal in concentrations of the solute. So if you look at the number of solutes per unit volume, you're going to see that they're equal in that way. So that's what we have in the right U-shaped tube. We have dynamic equilibrium. And what we say about the two halves of the right U-shaped tube is we say these two halves of the U-shaped tube are now isotonic. Iso means same. They have the same concentration of solutes. Now we can apply this to this concept of osmosis to cells. Whenever we give just pure water to plant cells, they'll take on water and they'll just become turgid. They'll be just full of water, because pure water is going to be hypotonic relative to the cell interior, the plant cell interior. So water's going to move into the plant cell becomes turgid, but the cell wall does not allow the plant cell to burst. Now, if you and I are given an IV of pure water, what that can lead to is, once again, that pure water is hypotonic relative to our red blood cells. Water rushes in and can actually pop our blood cells. So we're not going to be given pure water even though we're dehydrated. We're going to be given an isotonic solution of saline or glucose or something like that. That way, we don't have too much water rushing into blood cells and popping them. Now, if we were say stranded in a boat or something out in the middle of the ocean, which is 3% saline for the most part, we can't drink the ocean water because it's hypertonic. It has a greater concentration of solutes. Remember, that's what we have over here. A high concentration of solutes means hypertonic, and that's what we have in this case. If we were to drink ocean water, what would happen is we'd start pulling water out of our red blood cells, causing them to shrivel. And that can happen with plant cells as well. If you add salt water to plants, it'll pull water out of them, and it can certainly kill them. So, in the hospital, the IV that we're going to be given is not going to be pure water, and we cannot drink seawater safely. As I mentioned before, there's simple diffusion whereby nonpolar molecules can get across the hydrophobic zone, which is this area here, on their own. In many cases, they will just diffuse down their concentration gradient. But if we have a molecule that's carrying some charge, it's going to need help. And even like water molecules need some help. And here we have a channel protein that's providing some help. Now that's also being shown over here. This is a channel protein. It just has a channel way that protects the charge of the molecules from the hydrophobic zone of the bilayer tails. And then here we have a carrier protein. And the carrier protein actually binds to the solute, and then that will help bring across the solute. So in both cases, whether it's a carrier or it's a channel protein, we're having transport proteins help move materials across. Now, if it's a passive process, in other words, if no energy is being invested, if energy is not needed to get things done, then this is going to be facilitated diffusion. So we've already said that nonpolar materials can move across the hydrophobic zone with little difficulty, like oxygen and carbon dioxide. So we know the answer to this question. Why are transport proteins needed? Because ions and polar molecules have a hard time getting past the hydrophobic zone. Now, some transport proteins, as we've already illustrated here, are channel proteins. Now, they are specific for what they'll help get across a membrane. Now, one of the key channel proteins are the aquaporins. They help water get across. So that's a key channel protein, are the aquaporins. And we've already noted also that some transport proteins are carrier proteins. And one example of that is a glucose transporter being a carrier protein, and it's embedded in the membrane of red blood cells. All right, so we've taken a look at passive transport. Now we're going to be taking a look at active transport. Now, a key thing we must remember about active transport is that active transport requires the cell to expend energy, and it's going to involve the movement of a solute against its concentration gradient, which is the opposite of what we saw in passive transport. Well, let's take a look down here at the bottom of the screen. And what we see is we see four different kind of stages of active transport. Now, in the first stage, we have a carrier protein, which is what's going to be involved in all active transport mechanisms. And this carrier protein is open so that solutes from the cytosol can bind to its binding sites. And that's what we see happening in step 2 of the process. The solutes have bound to the binding sites of the carrier protein, and then what we do is we phosphorylate the carrier protein. Now, we do this by hydrolyzing a terminal phosphate off of ATP, and that terminal phosphate will be added to the carrier protein. Now, in doing so, we move over to step 3. That creates a change in the shape of the carrier protein, this phosphorylation event. Now, notice what's happening. This change in shape results in the solutes being moved from the cell cytosol to the cell extracellular fluid. In other words, the solute is being moved from inside the cell where it was less crowded to outside the cell where it's more crowded. That's what we're seeing in step 3. And then in step 4, so we can get the carrier protein ready to do this work again, we remove that phosphate off the carrier protein. That causes the carrier protein to resume its original shape. And now it's receptive for the binding of a couple of more solutes. So, what we've seen is that the solute was moved from, in this case, in this set of drawings, was moved from inside the cell where it was less concentrated, this is key, and the solutes were moved outside the cell where the solute concentration is actually greater. And this, once again, is the opposite of what we saw in passive transport. Another key feature of active transport that we've mentioned is that ATP is the source of energy to get this active transport work done. That's a second component. Active transport requires the expenditure of energy, and it's almost always going to be in the form of ATP. Now, what's key about all of this is that this kind of work enables there to be a difference between the cell interior and the cell exterior, and this is key. For instance, in the case of animal cells, we will find a higher concentration of potassium ions versus outside the cell. And also, inside the animal cell, we're going to find lower concentration levels of sodium ions and calcium ions. And this is important to the normal functioning of cells. So active transport, we're going to remember a few things, and that is, in active transport, energy is expended, usually in the form of ATP. That's number 1. Number 2, it involves a carrier protein. Number 3, it involves a solute being moved from where it's less crowded. Besides simple diffusion, facilitated diffusion, and active transport, cells also have bulk transport mechanisms by which they can move materials across the membranes. So what we're going to be looking at in terms of bulk transport, we'll be looking at endocytosis as a form of bulk transport and exocytosis. But each of these really involves large molecules being moved across cellular membranes. Now, we've talked about phagocytosis before. We talked about it involves something like a food particle, and we form kind of a pocket, a membrane pocket around the food particle, and eventually that pocket forms a vacuole around that food particle. Remember, we talked about merging a lysosome with that and breaking down that food particle. That's phagocytosis. Well, that's taking in a large molecule or food item. That's endocytosis. That's one type of endocytosis. Phagocytosis is. Another form of endocytosis is called pinocytosis. That's like a cell taking a drink. It's kind of like cellular drinking. So here we have a pocket forming again, and the cell's kind of taking a big gulp of the extracellular fluid, and that pocket will become a vacuole. Now, there's a third type of endocytosis that we're going to mention, and it's called receptor-mediated endocytosis. Now, in this case, specific molecules, like LDLs is one example, will bind to specific receptors, and when enough of those have bound, then there can be this whole coated pit that forms, and what then happens is this pocket forms, and the LDLs are taken up in that way. So these are all forms of endocytosis, which refers to the uptake of material. So phagocytosis is when we have a cell that's eating, and in this case that we gave an example of, we actually have, I believe this is an amoeba over here, and it's using its pseudopodia to engulf a food item there. And anyway, so we've already talked about how the food vacuole can merge with the lysosome, and then we get hydrolysis of the food item because of those digestive enzymes in the lysosome. And we also looked at pinocytosis, which is cellular drinking. And pinocytosis is used primarily for the uptake of just extracellular fluid, like a cell taking a big drink. We also looked thirdly at receptor-mediated endocytosis, where a receptor must bind to a specific ligand. In this case, this little purple triangle represents the specific ligand. And I gave the example of something like LDL. Low-density lipoproteins can behave as a ligand to help stimulate the cell to take up the LDLs. Now, what's interesting about this particular example is that some people don't produce receptors for LDLs, and so they can't run receptor-mediated endocytosis for LDLs, and so their LDL levels remain elevated. And this is certainly the case in familial hypercholesterolemia, where individuals lack LDL receptors, and so they can't bring the LDLs into the cells. Now, besides endocytosis, there's also exocytosis. Now, once again, this would be like a Golgi vesicle pinching off from the transface, merging with the plasma membrane. Like maybe say when we release insulin from beta cells of the pancreas to get the insulin into the blood. Those are examples anyway of exocytosis. Now, I started off chapter 2 with saying that living things are bags of chemical reactions, and since all living things are made of cells, that we can say cells are little bags of chemical reactions. Now, one of the things we have to keep in mind when we talk about chemical reactions is that in chemical reactions, what we have going on is just rearrangement of matter. We are not going to have any new atoms being created. We're not going to have any atoms being destroyed. So when you have, say you have breakfast, you eat some breakfast cereal, you're taking in a lot, plant matter, and what you're going to do in the digestion process is you're going to break the plant, molecules down into little subunits, into their monomers, and then what you're going to do though is you're going to then you're going to use those monomers to make the molecules that you need as your DNA directs. Now, one of the things we want to keep in mind, of course, is that there's going to be release of energy as we break down some of these molecules. We're going to capture some of that energy in the form specifically of ATP, and then we can use that ATP energy form to do the work of the cell. And that's what we want to keep in mind about energy. Energy is the capacity to cause work or to do work. Okay? Now, we've already talked a little bit about thermal energy or kinetic energy or intrinsic kinetic energy. And one of the things we want to keep in mind is that if we can get stuff moving, then we can get work done, because moving objects can perform work by transferring motion to other matter. So think about water moving through a dam. Work can be done as water moves through a dam. Electricity can be generated. Turbines can turn, generate electricity. Think about legs pedaling a bicycle, for instance. Moving objects can perform work by transferring motion to other matter. So we get things moving, we can get work done. So thermal energy is really a type of kinetic energy that's associated with atoms moving randomly or molecules moving randomly. Okay? Light is actually another form of kinetic energy. Now, there's also potential energy, and potential energy refers to energy that matter possesses as a result of its location or position. Now, one form of potential energy is chemical energy, and this is one that we'll be dealing with quite a bit. And chemical energy is that energy that can be used to get work done in a cell. And it's probably the most important type of energy that there is for living cells, living organisms. Now, in the study of energy transformation, that is thermodynamics, we have a couple laws we're going to be talking about. The first law of thermodynamics really has to do with energy in the universe is constant. It's not changing, okay? But it's, even though it's constant, it can be transferred, it can be transformed, talking about energy. But just keep in mind that the amount of matter, the amount of energy in the universe is constant, but it can be transferred or transformed. So let's talk about that just a little bit. Think about in your vehicle, your car. You put gasoline into your tank. You start up the engine. Of course, the engine requires oxygen. You get combustion of the gasoline. And so what's happening is the gasoline is put into your car in liquid form. And then when you have a chemical reaction occurring within the engine that changes the gasoline from a hydrocarbon, from different kinds of hydrocarbons, into carbon dioxide and water that are going to be coming out of the tailpipe. So we put a liquid fuel into the gas tank, and what we're getting out of the tailpipe is we're getting these gases. So there's been just transformation. But these are not new atoms. These atoms were the atoms of the fuel and the oxygen. So there's just been rearrangement of these molecules into these products. These products have less energy because as the fuel and oxygen were combined in the reaction, they were giving off energy, and some of that energy was used to get the car moving. Some of that energy is just lost off as heat. That's why the hood of the car gets hot. And so we're going to see a little bit later how in our cells we don't use gasoline, we use glucose and oxygen, but it's kind of much of the same kind of thing going on. We're going to rearrange the atoms of glucose and oxygen, and in the rearrangement, in the transfer of energy from glucose form to ATP form, we're going to lose some of we're not going to capture all the energy in glucose. Some of it gets lost off as heat, but we will capture some, and the waste products look again, so carbon dioxide and water. Now, in the second law of thermodynamics, this gets at recognizing that whenever, we've already said the first law of thermodynamics is that, the amount of energy in the universe is constant. And so what happens with the energy, it's transferred or transformed. And the second law of thermodynamics has to do with that as we have these energy transformations, then what's going to happen is we're going to lose some energy as just heat. And so this energy is really unavailable to do work. So low-grade heat, it just can't get work done. There has to be a great enough differential in order for heat energy to be able to do some work. So what the second law of thermodynamics really gets at is that every time we have energy transfers or energy transformations, then there's going to be an increase in what's called the entropy, the disorder or the randomness of the universe. So energy conversions increase entropy because some of the energy in what we started with will be lost as low-grade heat energy in the energy transformation. Now, when we look at chemical reactions, what we find is that chemical reactions are of two types. They either release energy or they store energy. Now, those chemical reactions in which we have energy, a net release of energy, we call exergonic reactions. So look what we have here in this bar graph. So as we move up the y-axis, we see that there's an increase. It's a measure of the increase in the potential energy of the molecule. So here in this particular bar graph, we see the reactants have, double the amount of potential energy as compared to the products. So what happens is we can imagine this, for instance, being like glucose during cellular respiration. The glucose and the oxygen are the reactants. They have a lot of energy about them, especially the glucose. But as glucose and oxygen are converted into carbon dioxide and water, the products, energy is being released. So that's an example of, say, an exergonic reaction. Now, that's what we find in exergonic reactions. The reactant covalent bonds contain more potential energy than the covalent bonds of the products. Now, the reaction is actually releasing energy to the environment. So, and the amount that's being released to the environment is actually equal to the potential energy difference between the products and the reactants. Okay? So, whenever we burn wood, energy is going to be released. Whenever we break down glucose, energy is going to be released. Now, we will get into the steps of cellular respiration a little bit later. But cellular respiration is a way in which we are releasing energy. We're going to do so in many steps. It's really kind of a slow burn of glucose, really, is what it is. Now, besides exergonic reactions, there are also endergonic reactions. Now, in endergonic reactions, there has to be a net input of energy. The reactants are actually poor in terms of their potential energy value relative to the products. So let's take, for instance, I think a good example of endergonic reactions would involve something like photosynthesis. The reactants are carbon dioxide and water, which are of low potential energy value. And energy from, say, the sun or just light energy is put into the photosynthesizer, so that carbon dioxide and water molecules are rearranged into organic compounds like a sugar or something. So energy must be absorbed during the reaction. There has to be energy being added to the system to get those reactants to higher energy products. And so once again, as I was saying, carbon dioxide and water with an input of energy become potential energy-rich organic compounds like sugars during photosynthesis. Now, living cells carry out thousands and thousands of exergonic and endergonic reactions because, once again, living cells are bags of chemical reactions. And all these reactions, all the combined reactions of all the cells of an individual, like the 10 trillion cells that make up you and I, make up the metabolism. So when we use that term metabolism, we're really referring to all the chemical reactions that occur within an organism. And once again, energy is going to be released as exergonic reactions are being carried out, and some of that energy will be captured and will be used to drive the endergonic ones. So that's how cells really couple the energy releasing and exergonic reactions with the energy requiring endergonic reactions. Now, oftentimes the coupling mechanism, though, is actually ATP. In other words, the energy from exergonic reactions is captured as ATP. Now, in cellular respiration, what we end up having is the production of ATP. ATP is really considered the energy currency of cells, because if cell work needs to get done, it's probably gonna require some ATP to get it done. Now, ATP stands for adenosine triphosphate. So here we have the adenine component attached to a ribose sugar, and together they make up the adenosine. And then there are 3 phosphates. Now, I know it shows a P. It's not a phosphorus, it's a phosphate group. Remember, that's a phosphorus surrounded by oxygens. And these 3 phosphate groups carry negative charge. They're not particularly stable out there because you have this clustering of like charges. But once again, we know something about the relationship between stability and potential energy value. If something is unstable, like gasoline, it has good potential energy. If something is stable, it has low potential energy value. So all 3 phosphates have a negative charge. They're clustered out there together, and so that makes ATP like a compressed spring. It's just ready to go off. Now, as I said before, the bonds among these phosphates are relatively unstable, and so we can carry out hydrolysis to break phosphates off the ATP. In doing so, once we hydrolyze the terminal phosphate, and that's what's being shown down here in the bottom part of our diagram. Here's the hydrolysis reaction, the addition of the water to break this third bond here. That's what we see here. And as that phosphate is hydrolyzed, there's a release of energy. And that really is an exergonic reaction, so energy is being released. And we know when energy is released, work can be done. Well, ATP, as I've said before, is really the energy currency of cells because it powers nearly all forms of all different types of cellular work. Now, the thing about it, though, is that we use a lot of ATP, so we have to be able to regenerate it, and we do so by way of cellular respiration. And what's interesting, for instance, if you're taking a weightlifting class, say you lift 8 or 10 reps in a set, and you may rest for 30 or 45 seconds, and then you'll do a second set. And actually, in that first set, maybe you've completely fatigued the muscle, and it can't really do another rep. But then when you rest your 30 or 45 seconds, what you're allowing is the muscle cells to regenerate some ATP. We can regenerate about 10 million ATP per second in a working muscle cell. Now, once again, what's critical in getting this done, of course, is we keep taking in high-energy food matter. Food matter that has quite a bit of energy stored in the bonds of the material. And as we digest, as we hydrolyze that food, energy is going to be released, which is exergonic. So that's what's being shown down here is the way we regenerate ATP from ADP in a phosphate is we have to have exergonic reactions that release energy that can drive the phosphate back onto ADP so that we get ATP being produced. So we can hydrolyze it and do work. So some of the energy that's released during exergonic reaction with the help of oxygen can be used to actually add a phosphate back onto ADP, phosphorylating it back to ADP. So to produce ATP, that's endergonic. So we have a coupling, once again, of exergonic and endergonic reactions. They are connected with one another. So one of the things we can now should be able to kind of answer perhaps is, what kind of changes are going on as we get into shape, so to speak? Well, we can actually increase our number of mitochondria in our cells, in our active cells, so they can produce more ATP for those cells so we get less fatigue, but also that in order for those mitochondria to work efficiently, there has to be plenty of oxygen being delivered, and so we can actually build blood vessels to those cells, more blood vessels, and that's part of getting into shape. Now, one of the things we can't forget in all of this, though, is that enzymes are needed in chemical reactions within cells, because what they do is they lower the amount of energy required to get a reaction to occur. So here we have a diagram. Here we have several diagrams actually illustrating that point. Here we have this reactant. We're trying to get to these products, but what do we have to do? We've got this wall we've got to get over, and so that wall is called the activation energy barrier, and so it's the amount of energy that's really kind of needed to destabilize the reactant so it will become these two separate products. But look how high the wall is when an enzyme is not around. But once we have an enzyme grabbing the reactants, then look how much energy is really needed to get the reactants to change. It's much less. So that's what enzymes can do. They can grab the reactants, that's what's being shown here, and they can actually add stress. They create these micro conditions that's conducive to the reactants becoming the products. So enzymes are critical. Now, it's not uncommon. As a matter of fact, it is common. It's what happens is that structures or molecules that are highly ordered tend to move towards disorder over time. I mean, you can think about the Roman Colosseum and how it's crumbling over time. So what happens in nature is that high energy systems, unstable systems, tend to move towards low energy states or low energy systems, towards greater stability, in other words. I mean, you think about our own sun. It's halfway through its life. It's moving towards greater stability. It's losing energy and particles every single moment of every day. Now, proteins and DNA and carbohydrates and lipids, they too are highly ordered. They are high energy compounds. So why don't they spontaneously just break down into low energy, disordered compounds? The reason they don't, once again, goes back to what we were talking about earlier. There is this high energy wall that prevents them from just breaking down rapidly. So this energy barrier must be overcome before you can start getting these changes. And that wall, that barrier, is called the activation energy. And what it really is it's where the reactants have become destabilized enough that it's conducive to changes occurring. So this is how we get highly ordered cellular molecules that we take in by way of our diet to be broken down. We have enzymes that lower this energy barrier, this activation energy barrier, lower the wall, so we can digest the food and get the release of energy from those high energy compounds. Now, we've been saying all along that we're bags of chemical reactions. So if these walls, these activation energy barrier walls exist, how can we be bags of chemical reactions? Once again, it's all about enzymes. And by the way, enzymes, they're proteins that we produce at the direction of our DNA. And once again, what they do is lower the energy barrier, lower the wall, contort the reactants into an unstable state, add physical stress, create electrical conditions to where the reaction's going to occur. The reactants are going to change. Now, one of the things that's really important about enzymes and remembering about enzymes is that it's not that they carry out one reaction and then that ruins them. They are not consumed as they catalyze reactions. Instead, an enzyme can carry out more than perhaps 1,000 reactions per second. So they're fast workers. They're workers that are dependable. They can just keep working. And once again, we build them at the direction of our DNA. Now, something else about enzymes is that they are specific for what they will work on. And what they work on are called substrates. So they're specific for what they will work on. Now, the way that an enzyme generally works, here we have an enzyme, and here we have like a little pocket or groove in the enzyme. We call that the active site. Now, the active site is the site to which the substrate can bind. Once again, there's a fit between the substrate and the active site. So not just any molecule will fit. Enzymes are specific for what they'll work on. And so here we have the enzyme. In this case, you can see it's contorting the bonds. It's adding some physical stress to this disaccharide. Water is added. Remember, this is going to be a hydrolysis reaction because we're going to actually break glucose and fructose apart as products. So the site to which a substrate binds is the active sites. And oftentimes, what we have, as a matter of fact, the name of this enzyme would be sucrase. It acts upon sucrose, the sugar. So oftentimes enzyme names will end in ACE, A-S-E. Now, there are environmental factors that are really important in, enzyme activity. Temperature really matters. If temperature is too low, then the collision rate between an enzyme and its substrate will be low, and so the product rate, or the rate of production of the products, will be lower than optimum. If the temperature is just right, then there will be an optimal number of collisions of the enzyme and the substrate, you get an optimum rate of products being generated. But if the temperature is too high, that can actually denature. That can cause the enzyme to change its shape to where it won't even work. So, higher than optimal temperatures can denature the enzyme. And what we mean by denature, that means change the shape of the enzyme to where it can't work. What an optimal pH or optimal temperature will do is have the enzyme in the right shape, and it'll work as fast as it possibly can. And pH is another one of those environmental factors that's really important to the functioning of an enzyme. So a non-optimum pH can actually denature, change the shape of the enzyme to where it can't work. It can't fit the substrate for which it was made. Now, an optimal pH for many enzymes will certainly be between 6 and 8, but that's not always the case. As a matter of fact, in our stomach, we have an enzyme that works upon proteins. It's called pepsin, and its optimal pH is 2. And then when our acid chyme, the food from our stomach, goes into the first part of our small intestine called the duodenum, then the optimal pH for enzymes there that are going to be involved in the digestion of the food is about 8. So pepsin's not even going to work. But there will be other enzymes that will take over for pepsin there in the small intestine. Now, some or many enzymes actually require cofactors to either bind to the enzyme itself or even to the substrate. And some of these cofactors include like zinc ions, ions of iron and copper as well. So that's why on like your multivitamin you might see, or even on some of your cereals, you'll see that zinc or iron or copper has been added. Is for that reason. These are cofactors. Now, some of our vitamins are also cofactors, and we call them coenzymes. For instance, folic acid is a coenzyme for many enzymes that are involved in the synthesis of nucleic acids. That's why it's so critical that we get plenty of folic acid, or that, let's say you have an expectant mother, she gets plenty of folic acid, is because that synthesis of nucleic acids is so critical to the normal development of a fetus. Now, we have to be able to control enzymes. We don't need them on all the time, and we've got to be able to turn them on and off as needed. So we control enzymes through inhibitors. Now, what's being shown down here is when, say, a substrate is about to bind to the active site, but also notice there's this little pocket here on the enzyme. It's called an allosteric site. Now, so I'm just pointing out kind of the anatomy of an enzyme. But in some cases, there are molecules that can bind to the active site other than the substrate, and they are called competitive inhibitors. They bind to the active site. That's key. They prevent the substrate from binding. That's key. So, there are competitive inhibitors that can bind to the active site of an enzyme and block the binding of the substrate. But in other cases, molecules can bind to that allosteric site, this other little pocket, and when they bind there, that changes the shape of the active site so that it no longer fits the substrate. Now, that's called non-competitive inhibition. When the molecule binds to a site other than the active site, but it still changes the shape of the active site to where the substrate cannot bind to the active site of the enzyme. Now, there's also something, I want to show you another way in which enzymes are controlled. Here's a pathway that illustrates threonine, which is a type of amino acid, being converted into isoleucine, another kind of amino acid. So threonine binds to an enzyme called threonine deaminase, and threonine is converted into an intermediate, which is acted upon by a second enzyme, which converts the first intermediate into a second intermediate, which is acted upon by a third enzyme, and so on until the final product, isoleucine, is produced. Now, when a cell is using isoleucine up, then that's fine. But once a cell has been told, or it understands that it no longer really needs to build that particular protein, or it's not needing isoleucine as much, then isoleucine will actually start binding to the allosteric site of threonine deaminase. And when it binds there, it changes the shape. Let's see, I guess this is the drawing to look at. When isoleucine binds to the allosteric site, it changes the active site shape in such a way that threonine can now not bind to the threonine deaminase. And so isoleucine is shutting itself down, its own production down, essentially. That's called feedback inhibition. Now, many drugs, pesticides, and poisons are actually enzyme inhibitors. So, there's the story of sarin gas being released in a Tokyo subway back in the mid-90s. Well, that sarin gas shuts down an enzyme called cholinesterase and acetylcholinesterase, and that enzyme is critical to the normal functioning of the nervous system. And so the nervous systems of those individuals that breathed in the sarin gas were no longer functioning like they should have. They were not under appropriate control. So many individuals died from that.